and its regulation. Cell cycle means when our cell, we are talking about the eukaryotic cell, means any tissue cell. Suppose you have cervical, muscle cell, liver cell, bone marrow cell, blood cell, lymphocytes. So all these cells retain DNA material, double stranded DNA material, that is 46 is number, and it requires a very regulated manner duplication. So for this, the cell has evolved a very tightly regulated interphase and M phase in the cell cycle. So it's a regulated manner. Cell divides itself and produce two new cells. So here is the one cell and here the offspring two cells. These are the two newly formed cells, 1 and 2. But this cell again need to divide. So for that we have interphase where gap 1. This gap 1 phase nothing but cell will assume that all the cell organelles has formed once again, and ribonucleic acids are mRNAs are forming, and the size is continuously increasing. That means all the nutrient and everything should be good enough to move in the S phase. So what is S phase? As you know, the name is synthetic phase. Synthetic phase, S-Y-N-T-H-E, synthetic phase. In this phase, in this eukaryotic nucleus, it actually creates multiple origin of replications, and this origin of replication along with this 3' to 5' and 5' to 3', this will act as a template strand, and as it is in the as it follows all the rules of the eukaryotic replication, and it will form towards the continuous synthesis, that is leading strand, and away from the form the lagging strand. So this lagging strand will incorporate this 200 base pair Okazaki fragments along with the RNA primers I have shown here in the blue dots. So today our point is only to discuss the cell cycle. So here you can see this complementary nucleotides, here A and it is T. But if you see the next consecutive base pair, you will find that D-ATP is the rule. It's complementary rule, so it's required D-TTP. But if some misincorporation occur, so it is a fault in incorporation. So you will find that after the S phase, during the S phase, any wrong DNTPs is incorporated, it is error in the replication, so it will act as a hindrance to progress of this cell cycle. So it will act as a stop signal. So this is not only that, before that we have discussed that if the G1 phase, if do not find sufficient nutrient and the cell do not grows as it should grow enough, it's enlarged in size, volume. If it is not, do not find sufficient nutrient, so this G1 phase will move into G1 to G0 state. So this is another. And you see, after this replication has been completed, so cell will move to the G2 phase. But before that, your 2C content of this DNA is now 4C, and in the middle of this chromatin there is some kinetochore or primary constriction. So this is for the spindle assembly checkpoint, and it will attach with the chromosomal fiber of microtubule, and this microtubule will move from here. So now you can see After the S phase, G2 phase, we once again see M phase prepare to start mitosis. That means it will divide the cell. DNA synthesis is good enough or it has no error, it will confirm once again. RNA synthesis of RNA, that is transcription, will take place in this cytoplasm. You can see the DNA will move and here the mRNA, and it will attach some ribosomes, and this ribosome will synthesize new protein from the amino acid or polypeptides. So now our discussion is that what actually assures the cell that it has no error in its whole process. That means G1 phase, G2 phase or S phase. During these three phases there is no error. So for that the cell has created some scrutinizing or surveillance system. So this surveillance system is called checkpoint. So if we discuss one by one, what are these checkpoints? So these are G1 restriction checkpoint, S phase checkpoint, and G2 checkpoint, and another is APC, anaphase chromatin contrast checkpoint or spindle assembly checkpoint. So as I have discussed that G1 restriction checkpoint will check that whether the cell has grown all its cell organisms, that is centriole, mitochondria, Golgi body, endoplasmic reticulum, all have followed the normal C. So it is, if it is not met, then it will move to the G0 state. And we know that the nerve cells is on G0 state, and there are several cells in the eye also you will find. S phase, where completion of the duplication of the DNA double strand, and it is, if it is DNA, so DNA have alpha, delta, epsilon, gamma and zeta. This will check, they will synthesis primer, this will synthesis probably, this will synthesis the primer, and these two probably delta and it will synthesize leading and lagging strand. I will discuss it in another video. But for what is the completion of DNA duplication? So these are the enzymes that require the to complete the DNA replication. As you have seen, 2C2, 4C state. So APC checkpoint, I want to elaborate this thing. If you see the centromeres, the microtubular organism center, where you have centrioles, and from here some spindle moving out, and this will attach to this kinetopores. So in everything, whether it is anaphase A or anaphase B, it will detach in the middle, and the chromosome will move towards the end, and after that the reappearance of the cell nuclear membrane. So it will assures you a completion of cell cycle where one cell divides into two. But the key mechanism, that is how this regulated manner assures you that this has happened or followed all the processes. So for that the cell has evolved some MPF factor, mitosis promotion factor, where two important proteins, one is simple protein, that is cyclin protein, cyclin D, E, A and B in respect of time I have given, and another is cyclin dependent kinase, CDK protein. So CDK does one thing. During the G1, S, G2, M phase, we will see that according to the phases of the cell cycle interphase, that is, it is a gap phase. So it is a time span. So where you will see this CDK4 and CDK6 cyclin will combine with these two, and they will phosphorylate some enzymes. So phosphorylate threonine, or it may be tyrosine, threonine or tyrosine. These two is being these two amino acids in the substrate get phosphorylated with this thing. So phosphorylation means it will active the machinery, and after the thing the cyclin will destroyed by some proteasome. So for you I can give you that in the G1 phase cyclin D combined with CDK4 and CDK6, and cyclin E in the S phase, and of course in the G2 cyclin A, but they will combine with same CDK2, and they will assure the all the that means signal that helps to move forward or to complete the cell cycle or cell division. So CDK1 in the M phase, which will assure whether the spindle assembly checkpoint has worked sufficiently at this. So in this way, if everything goes fine, CDK1 will show transition from G2 to M, and I have already discussed G2 to M transition. So APF, cyclin and CDK complex, and cyclin D, CDK, cyclin dependent time. So it depends on this cycle. If both remain intact, they will work in the cell cycle throughout. Thank you. So in this way, it control the whole cell cycle regulation.